let's talk about the eyeball. So, light, important to see, like the birds. So what happens exactly? Light comes in. Let's say we have a light source. It's the sun because you're staring at the sun. Not a good idea, but you're doing it. So what's happening? Well, light's coming in from all directions. And then, first thing that's going to hit is your cornea. Yay, corneas are good. You wanna know why? It's because they're like this invisible film thingy full of crystallines that redirects the light so it's going straight on. Then it's going to hit a very important function or structure called your lens. Lens is a biconcave structure, meaning it's bowed out like that. Now, when it hits your lens, that is going to refocus the light, so the vast majority of it is going to hit this red structure in the back called the fovea. Fovea is a region of high acuity, really high photoreceptors. There's nothing else in there besides photoreceptors. Pretty cool. Neat those. All right. Now there is an important function because as we get further or closer, it requires us to change the angle at which light is bent. Point being, if this were further away, this is gonna get messy to the way I'm drawing this, but the light's gonna come in and that's gonna require it to bend less versus if it was really close, light's gonna come in and that's gonna require it to be bent more. And then at the lens, I guess, is where the bending is really occurring. In order to do this, our eyeball, the lens particularly, the ciliary bodies, so the lens is connected to something called ciliary muscles, which is housed inside the ciliary body. And our lens, if this is the biconcave structure, can go like this or like this. So light is hitting a more flat thing or a more bowed thing. The more bowed thing allows light to be bent further, and a more flat thing allows light to be bent less further. Sure. That being said, when our lens needs to be contracted or needs to be squished, our ciliary lens have to contract and cause our lens to bow out like that, because that takes a lot of pressure. It's not what the eye normally wants to do. It's a lot easier to go flat. That's why we get fatigued when we're staring at our phones for too long, or reading a book, but who does that anymore? So as you can see, light comes in, hits the fovea, and in the fovea we have things called photoreceptors. We have two types of photoreceptors. I'm going to draw them out here for you so you don't get confused. I'm going to draw a simplified version because I don't necessarily need all the stuff on the bottom. There's more to the structure down here, but I'm not too worried about that. So, our things that are not a nice rectangle are called our cones. Yes, that would make sense. And then these things that look like rods are called rods. Cones, in particular, see color. And they also are responsible for acuity of our vision. And we'll talk about that in a second. Rods, however, they only see black and white colors. And these function in low levels of light, where these function in high levels of light. So when it gets dark, we start to lose the ability to see colors and things. It's because our cones are essentially turned off while our rods are super active. And they're like, ooh, my turn. I gotta see stuff. Cool. So that is the story of how we see stuff, or how light is bent and stuff. Now we need to talk about this diagram over here. It's a little bent, but I think it'll do. Nah, I don't like it. Yeah, it'll work. Okay. So, right here we have this protein, and when light comes, so this represents a photoreceptor. Could be any photoreceptor, but in general it's a photoreceptor. So we have a light particle that comes in, squiggly line means light, and it's going to activate, or it's going to hit this protein complex that has retinol in it. Now we're going to go from a retinol that's a cis version to a retinol that is a trans version. Now, when that happens, something very important happens. We have a subunit up here called alpha. Now, there's also a beta and a gamma subunit, but we're not going to worry about those because the alpha subunit is actually going to be what detaches and it's going to activate PDE. Now, what's PDD? PDE mean? PDE, for reference, equals phosphodiesterase. You want to know what that is? Go ask someone else. Google. Sorry. PDE will then in turn act on our cyclic guanine monophosphate and decyclitize it and turn it into just good old guanine monophosphate. That in turn is going to erase this connection that we had to this ambiguous gate, and this gate is now actually going to open. Now this is a cationic gate, meaning anything can flow in. So we're going to have both potassium and calcium are going to flow in, but sorry, I meant sodium. Sodium and calcium are going to flow in, but mostly sodium, and in turn, they're actually going to cause a Bourn on this calcium voltage gated channel. In reality, it's probably closer to where we're going to be at, but for space's sake. Now that's going to open, and that calcium is going to go in and react with my neurotransmitter over here, and glutamate's actually going to began to be released. So what happens when glutamate's released, or it starts to come out, glutamate's released, and then it's going to come and attach to our mGluR6 receptor. All right, now once our phosphodiesterase is activated, it's going to come over to our protein, or to a molecule right here called cGMP, central cyclic guanine monophosphate. And that is actually going to essentially knock the C off. Not really, it's actually C stands for cyclic, so we're decyclitizing our molecule, but that's going to cause it to fall off and become GMP. But likewise, cGMP was keeping this cationic channel open. Now it's not doing that anymore. So this cationic channel is going to close, meaning sodium and calcium can't come in anymore. So we erase that path, and then, oh crap, so their Bourn was keeping open our calcium channel. But now that's going to close. Oh no. This string is really falling apart, isn't it? Poor cell. Oh crap, but not calcium was causing glutamate to be released into our extracellular environment. But now that's not going to happen. Wah wah. Now there's two pathways that I erased here. I want to talk about each one independently. So we're going to start with this bottom one. This is the on-center route, and we'll talk about more what that means later. On-center route. So before, glutamate was staying attached to this mGluR6 and sort of inhibiting it, if you will. But now that it's not, mGluR6 is actually going to come in and through some mechanism open up this channel, which is a calcium channel, specifically calcium, and that is going to open up another channel and allow our neurotransmitters to come here. Something I forgot to do that I should probably do is we need to give these guys names. So like I said, this is the photoreceptor. It's receptor, you get the idea. This is a bipolar cell. And this is a ganglionic cell. And that goes to brain once it's excited. So woo, keeps on going down. That's fun. And this is, I don't know for sure, but I suspect this is why it's called a bipolar cell. So in some bipolar cells, this is what happens. You know, the deactivation of our photoreceptor actually causes an action potential of the bipolar cell. However, other bipolar cells, in which we call them off-center cells, because glutamate was actually activating our AMPA channel, well, crap, no more glutamate. So that means no more ions, which means if we had a channel open before, we are actually going to now close that channel. So when we inhibit the photoreceptor from depolarizing, on an off center we also inhibit an AMPA channel from depolarizing, and therefore we don't get a signal to the brain. Now, a combination of this on-center or off-center idea of sometimes they cause light, or sometimes they cause signals, sometimes they don't cause signals, actually ultimately the reason why we can make out like hard lines and shadowing and depth, because our brain, the way the light is hitting our brains, allows us to see like, oh, hey, this marker has an edge to it because look how much light is bouncing off here, but there's not enough light bouncing off here. In that sense, darkness is almost kind of like a signal in of itself. Weird to think about, but that's how it works. We also need to talk about the on. All right, I want to talk about our on-center, off-surround, this guy, versus our off-center, on-surround, this guy. So I've made up a picture here. I might need to draw a separate one here in a minute, but we'll see how this one goes. So what we're talking about is what on center means is sort of like that picture. That picture I drew earlier with the huge neuron pathway. There's also a second pathway called the surround pathway. That essentially we have our rod. So let's say this is our photoreceptor, and that's actually going to synapse to a horizontal cell. What's a horizontal cell? Well, they actually act to release GABA. So that's GABA, and that's going to influence another photoreceptor cell that directly synapses to our bipolar, which then synapses to our ganglionic cell. So what GABA does is it actually causes an IPSP in this neuron here. So when we're releasing GABA, we're actually decreasing the effect, if that makes sense. So erasing, well, actually I'll keep that there for a sec. We might need that. So when we have a light particle that comes and hits our on-center cell, we're going to go from the point where we've already released we stopped releasing glutamate into our extracellular environment. Because no matter whether it's on-center or off-center, notice that the changes only occurred in the bipolar cell. They didn't occur, everything before that on the photoreceptor was perfectly normal. So on an on-center cell, what occurs is we can use a rule saying that when we're on something, we have an mGlu-R6 channel. When we're off something, we're using a AMPA channel. So that'll help us keep straight of what are we talking about, what's happening when. All right, so on center talking about we're not going through the side pathway, but we're going directly to the bipolar cell itself, when we're using an mGlu-R6 pathway. So light hits it, and that's going to cause less GABA to be released, less glutamate to be released, so less GLUT to be released, and that will in turn activate the MR6, or activate MR6, that should be the GLU MR6, which will then allows calcium to enter, which will then cause the ganglia cell to depolarize. If light is hitting our off-center on surround cell, or the off-center portion, well, again, that will still be less glutamate, but that is going to, now we're talking about an AMPA channel. So what, if we think back to our diagram from before, When we remove the glutamate, we're actually closing a channel. We're closing our AMPA channel. So, close AMPA. Which will then, if you think about our pathway, so now from here to there, we're no longer releasing glutamate to stimulate our horizontal cell. So if we're not stimulating a horizontal cell, then we're not releasing GABA. So we'll say horizontal, almost stopped at HOR, not stimulated. Then GABA, not released. So, is that going to help us depolarize the bipolar cell, or is that going to make it worse? Well, it's actually going to help us, because if GABA was released, then we would have more influence. But if GABA was released, then we would actually be down-regulating this whole process. But because GABA isn't released, we're going to say ganglia depolarized. Okay, now we're going to take that idea and come over to our off-center on surround. So now we're still talking less glutamate, because that's the name of the show. Less glutamate. Cool. What happens then? Well, we're going to close the AMPA channel. If we're closing, sorry, I forgot my photon. If we're closing an AMPA channel, then what's not happening? Well, we're not depolarizing. Not depolarizing bipolar, which means no ganglia stimulation. Sad day. That sucks. But there's hope. If our light is hitting our positive, or rather our on surround off center, then what we will see is we're still seeing less glut, but we're now activating GLU MR6 or something like that. Then that will in turn cause us to that'll cause depolarization on horizontal, which will cause release of GABA, which will ultimately cause a downregulation. Less ganglia stimulated. Okay.